Hello, Elmira is with you again, and today we will talk about modern methods of qualitative and quantitative analysis of biologically active substances in plant materials. In the first part of the lecture, we will discuss the classification of analysis methods, and in the second part, we will consider the use of qualitative and quantitative analysis methods on certain biologically active substances. In chemical analysis, it is first of all important to make a difference between qualitative and quantitative analysis. The task of qualitative analysis is to determine the qualitative composition of a substance or mixture of substances. That is, qualitative analysis answers the question, what is it? Chemical qualitative analysis is based on a reaction that is accompanied by an external effect. For example, a change in color of the solution. The substance used for analysis is called a reagent, and a number of requirements are presented to it. First of all, selectivity. Quantitative analysis answers the question how much of it, and can be carried out by a wide variety of methods. The methods of analysis used to determine biologically active substances can be divided into two groups, classical and instrumental. In some countries, for example in Russia, we divide the methods of analysis into chemical, physicochemical, and physical. But the division into classical and instrumental is simpler, and this will be enough for us to have an idea about the methods of analysis. An analytical signal in chemical methods of analysis is the result of a chemical reaction. In instrumental methods of analysis, the analytical signal is usually recorded using special equipment. These include, first of all, optical spectroscopic methods of analysis based on the ability of atoms and molecules to emit, absorb, and scatter electromagnetic radiation. Registration of the totality of received signals makes it possible to characterize its qualitative and quantitative composition. These methods are based on a quantitative study of the relationship between the composition and physical properties of an object. Let's start with a consideration of classical methods of analysis. In the classical definition, three stages of analysis are distinguished. The isolation of biologically active substances, we talked about these methods in the last lecture, qualitative analysis, and quantitative analysis. Qualitative analysis is usually based on some kind of chemical reaction, such as precipitation, formation of a colored complex, evolution of gas, or pyrochemical reaction, such as the coloring of a flame by certain metal ions. For example, everyone knows from school that sodium cations color the flame yellow. A qualitative reaction must meet a number of requirements: high speed, the presence of a characteristic external effect, irreversibility, high sensitivity. The main classical quantitative methods in analytical chemistry are titrimetric and gravimetric. The titrimetric method of analysis is based on measuring the volume of a reagent solution of exactly known concentration consumed for the reaction with the substance being determined. The law underlying it is the law of equivalence. For analysis, a solution with a substance with an unknown concentration is placed in a flask, a suitable indicator is added, and titrated using a solution with a known concentration, a titrant. The concentration of the analyte is determined by the volume of the spent titrant. The gravimetric method is highly accurate and is based on determining the exact mass of a substance. This method is used as a rule for the determination of mineral substances, since high temperatures are used in the analysis process. Instrumental methods include a large number of different methods of analysis, which are based on various physical, physicochemical, and electrochemical processes. The main groups of methods used to determine biologically active substances are spectroscopic methods based on the interaction of the analyte with electromagnetic radiation, chromatographic methods based on the distribution of the substance between the mobile and stationary phases, and electrochemical methods based on the use of the electrolysis process. In instrumental methods of analysis, qualitative and quantitative analysis as a rule are carried out simultaneously. Therefore, in this case, we distinguish two stages of analysis. Consider the using of methods on specific biologically active substances. Ascorbic acid or vitamin C is a well-known biologically active substance with a number of proven physiological effects. In the presence of oxidizing agents, ascorbic acid is readily oxidized to dihydroascorbic acid. This is the basis of its antioxidant action. Consider the analysis of ascorbic acid by classical methods. The qualitative determination of ascorbic acid is carried out using the following reaction. The first one. Silver nitrate is added to the substrate with ascorbic acid and the dark silver precipitate is formed. The second one is discoloration of an iodine solution when ascorbic acid is added to it. Quantitative analysis of ascorbic acid is carried out by the titrometric method, which can be based on both an acid-base reaction and redox reaction. Acid-base titration is based on the interaction with an alkali with a known concentration, a titrant. The analytical signal is the appearance of a stable, pink color of phenolphthalein present in the mixture. The concentration of ascorbic acid is determined by the volume of spent alkali, through which its mass is calculated. The unknown concentration of ascorbic acid is determined using the law of equivalence. Ascorbic acid, as mentioned earlier, is easily oxidized to dihydroascorbic acid. For redox titrations, potassium iodate is usually used as a titrant. The endpoint is determined by the reaction of the first excess portion of iodate with the iodide ions present in the solution, which leads to the formation of free iodine, which turns the starch blue. As noted above, instrumental methods allow both qualitative and quantitative analysis to be carried out simultaneously within a single experiment. When determining ascorbic acid by the spectrophotometric method, we will observe an absorption maximum at a wavelength of about 260 nanometers, which corresponds to the ultraviolet area of the spectrum. For quantitative analysis, it is necessary to obtain a calibration curve for a standard solution of ascorbic acid. If we carry out an analysis by the method of infrared Fourier spectroscopy, we will observe the following picture. IR spectroscopy is based on the interaction of infrared radiation with the substance under study. When infrared radiation is passed through a substance, vibrational movements of molecules or their individual fragments are excited. In this case, a decrease in the intensity of light transmitted through the sample is observed. Thus, we observe a pattern with several peaks, where each peak corresponds to a specific fragment of the molecule. To evaluate such spectra, libraries of IR spectra are used. Mass spectrometry is an analysis method based on the ionization of molecules of substances, the separation of the resulting ions, and their registration. Mass spectrometry is a highly accurate method of analysis that allows you to determine the molecular weight of a substance, the formula of a substance, and its structure. As a result of the analysis, we obtain a mass spectrum, which is a dependence of the intensity of the ion current on the ratio of mass to charge. The molecular weight of ascorbic acid is 177. Weaker responses are associated with fragments of the ascorbic acid molecule. Consider the analysis of such an important group of phenolic compounds as flavonoids. Phenolic compounds are mainly analyzed by instrumental methods. At the first stage, it is necessary to choose a standard reagent. Quercetin or rutin are usually used for the analysis of flavonoids and obtain its spectrum, which is used to determine the absorption maximum. Next, at this maximum, the optical density is measured for a series of standard solutions with a known concentration in the range under study, and the calibration curve is built. Next, the optical density of the test solution is determined, and the concentration of flavonoids is determined using a calibration curve. For example, if the optical density of the analyzed sample was 0.25, then the concentration of flavonoids in terms of quercetin is approximately 4.8 microgram in milliliter. Further, individual flavonoids can be analyzed by high-performance liquid chromatography. The method is based on the different response time of each flavonoid. On the chromatogram, we will get the peaks of individual flavonoids. The amount of each flavonoid in the HPLC method is determined by the area under the peak. In our laboratories, we carry out not only HPLC analysis, but also GC analysis, in which gas is used as the mobile phase. In the picture, you see chromatograms for organic acids and amino acids. Gas chromatography is used to determine volatile substances, but techniques such as derivatization can also determine non-volatile components. Well, I hope that in this lesson you got an idea of what the chemical analysis of biologically active substances looks like, and you can use the information for further work. Thanks for your attention.